it's my pleasure to introduce Ammon Corl, today's speaker. I think many of you know Ammon, but some of you may not, so I'm going to tell you a little bit about his history. Ammon did his undergrad at University of Arizona, and which was Michael Nachman's old stomping grounds. I don't know if you guys actually knew one another in those days.
We met.
Yeah?
No?
It's been many years.
We didn't interact much.
Yeah. But they're interacting now. Which is good. So, and then he did his master's at Stanford, and then he did his PhD at UC Santa Cruz, where he was working with Barry Sinervo. And he was doing his PhD work on the rock, paper, scissors game that he's going to be talking about today. After he finished his PhD, he and I actually tried to get him here on a postdoc. He wrote grant proposals to like UC presidents and such, but those things didn't work out, unfortunately. And he ended up doing a postdoc at Uppsala University working with Hans Ellegren. And that work has actually resulted in some really nice papers that have appeared recently, a couple of them. I don't know if there are more on the horizon, but these were papers using shorebirds as a model system. And when that postdoc ended, he didn't have funding for an additional postdoc, so he came back to the Bay Area, where his intention was to, basically write papers from home, write grants, that sort of thing, and make things work. And just by chance, he happened to be visiting here. Allison Davis, my former postdoc, was also a graduate out of the Sinervo lab, and Allison and Ammon are good friends, and so Ammon came by to visit. And I overheard that he didn't have a place to do his, to write, that he was just going to be writing from home. And I said, man, I can't offer you any money, but I can offer you a place to work in a rich intellectual environment here in the MBZ, which he took up, thankfully. He took that opportunity. And it's really, I'm going to tell you a little bit about this because it's been a really great, I think that it's been a really nice story, the way things have actually played out. Once he arrived here and started working, he was initially working primarily on the shorebird stuff. He became interested in doing some next-gen sequencing work oriented around extending the rock, paper, scissors game, looking at the genomic basis for it, and had a discussion with Rasmus, which ended up resulting in a joint project with Rasmus that Rasmus could help fund. And that project, as it took off, essentially became folded into a grant proposal that Barry Sinervo was writing, which provided him with postdoc funding, so now he actually has funding here. And so it's been great for Ammon, obviously, to have showed up at the MBZ and had this thing happen. But it's also been really great for me and for the members of my lab, because he's been a really incredible sounding board for us. I mean, we talk science, I would say, every day. Yeah. He's in the office next door to mine. I'm constantly talking to him about things. We've collaborated on a hummingbird project. And since he's had a chance to work with KB, which are genomics guru, as he's been absorbing that information, he's been playing that information back to the students in the lab, helping them with their projects. I mean, it's really worked out better than I ever could have possibly hoped when I said, hey, why don't you just take a seat in the office next door? So Ammon today is going to talk about his dissertation work, and I think at the end he may talk about the genomics project that he's initiated since he's been here. Thanks, Ammon.
Thanks so much, Jim. And
yours.
Thanks all of you for coming. I'm really excited to tell you both about what I did for my PhD, but also what I'm doing here, in case you haven't known or have only heard parts of this. So I'm going to just jump right in. Make sure these lights. So the big question that I've been investigating for a while now is a phenomenon that's confronted many different biologists, which is that within a single population of a species, you can see sometimes markedly distinct forms. So we have different lizard colors, different snake colors, different fish colors, different butterfly colors, birds. And so you can have polymorphism within a population that's strikingly different. And it's not restricted to just coloration. I also wanted to point out that plants can have morphs. There's heterostyle morphs that deal with mating isolation and things like that. It can be body size, it can be behavior, it can be a whole suite of characteristics. But this is a really interesting phenomenon, I would argue, is that you can have these specialized or markedly distinct types within a species, which is something that many people wouldn't necessarily think could happen. And there's been a whole body of literature about the maintenance of these, why they're around, all those sorts of things. That's a whole separate talk. But what I want to concentrate on is one aspect that many people have thought of when they've seen this phenomena, is that potentially polymorphism might lead to new species. And this is like an old idea, and it's driven by a few basic observations. The most simple one is the observation that morphs within a population sometimes show phenotypic differences to the same degree as different species. And so a really interesting example of this is actually there's a population of finches on the Galapagos where they have different bill morphs and they interbreed, though there's some sort of mating. And these bill morphs are sort of akin to the differences you see among populations or among species of Darwin's finches. Another reason why you might think that polymorphism might play some role in speciation is that sometimes you see species that are monomorphic, so they're not polymorphic at all, but they resemble one of the morphs in a closely related polymorphic species. And this suggests that, hey, maybe the same process, you go from polymorphism to monomorphism, and this can drive speciation. And lastly, even within a population, sometimes morphs can exhibit assortative mating and mating preferences, and even genetic incompatibilities. So a great example are the birds down at the end, the Gulvian finches. There's been a lot of great work done on these, and they show both assortative mating and also post-zygotic reproductive isolation between morphs. And so all these factors suggest that potentially polymorphism can have some role in speciation. And the question I'm going to ask in this talk is, could this polymorphism essentially be the starting material for new species? If I'm going to give a speciation talk, I'm going to give a very brief background on what is a species. How I like to think about species is a definition by Kevin de Quiros, is that species are independently evolving evolutionary lineages. And one way you can assess that is the biological species concept, in addition to other ways, which suggests, the biological species concept suggests that you want to look at interbreeding between populations. And if you have reproductive isolation between populations, those are separate species. And a common view of how speciation proceeds is that you have adaptation with geographic isolation. And this in turn eventually leads to incompatibilities among populations. They can no longer interbreed, they're separate species, all is good. And this, of course, is a dominant mode of speciation. I'm not arguing against that, but what I wanted to ask was, could the phenotypic diversification that we observe within populations in sympatry then be relevant for speciation, whether by allopatry or another mechanism? All right. There's a paper by Mary Jane West Eberhard who actually had thought deeply about this question and was thinking about polymorphism and speciation. And this actually was a good inspiration for a lot of the things that I did in my PhD. And so I just want to lay out a distillation of her theory. And first of all, she proposed that you get these polymorphic types due to strong interspecific competition with a population. That basically this leads to specialization on different morphologies or behaviors or what have you. However, once you have this variation, natural selection or sexual selection can act on it. And a particular morph can be favored in a different environment. And this can lead to the loss of a morph from a population. And this essentially changes the competitive environment. If the morphs arose due to competitive interactions among each other, taking one away changes that competitive environment, and this could lead to rapid evolution of the remaining morphs. And then lastly, you can imagine that populations containing different number of morphs could be reproductively isolated if the traits that are adapted to one competitive environment differ from the traits that are good in the other environment. And so I use this theory to generate some hypotheses that I could test with my data, and leading from that theory. So first of all, and this is going to be sort of a guide through my talk, so I'm going to address each one of these hypotheses. And first of all, you might hypothesize that if polymorphism is actually resulting in new types, that polymorphic types fix and persist to become species, then monomorphic lineages should resemble the morphs that are found in polymorphic lineages. If it's actually polymorphism that's promoting diversification, then polymorphism should be ancestral on a phylogeny, and monomorphic lineages should be derived from those polymorphic types. And then if the loss of a polymorphism actually resulted in a new competitive environment, maybe we can see evidence of rapid phenotypic evolution. And lastly, populations differing in the number of their alternative strategies could potentially develop reproductive isolation. All right, so that's the background, and now this is the star of the show. This is the side-blotched lizard, so named because of this big black dot on its side. A quite charismatic lizard, if I've ever saw one. And one of the things that's been made the side-blotched lizard famous is that Barry Sinervo, my PhD supervisor, discovered that they are polymorphic. So they have individuals with orange, blue, and yellow throats, and these correspond to different mating strategies. So the orange-throated males are more aggressive than the other types, have higher testosterone levels, and try and control large territories with lots of females. The blue morphs, in contrast, guard their mates more closely, so they have smaller territories with fewer females. And lastly, there's the yellow males, which are sneaker males, which aren't so territorial. They actually mimic female coloration. And this system is an evolutionary analog of a rock, paper, scissors game. So the orange ones can beat the blue because they're more aggressive. The blue can beat the yellow because they guard their mates. And the yellow can beat the orange because they can sneak onto the large territories of the orange males. And so over time, these morphs cycle in frequency over time, just like a rock, paper, scissors game. And just to give you a little background about the genetics that govern these mating types, Barry has named the gene that gives rise to these types the OBY locus, which just stands for orange, blue, and yellow. And it's theorized that this locus is a single gene with 3 alleles or 2 tightly linked genes, or something akin to that. And that you can observe 6 putative genotypes in the field that would correspond to 3 male mating strategy phenotypes. And so just to give an illustration of this, if you have a throat that's solidly covered by orange, we'd call that an OO, homozygote. Whereas if you have blue with orange surrounding it, that would be called an orange-blue heterozygote, and so on. And any individual with orange, we think that the orange allele is dominant. This gives rise to a more aggressive lizard. Whereas if you have sort of blue, dark blue covering most of the throat, we would call that one of the mate-guarding lizards. And since the later part of my talk, my focus here at Berkeley is on the genetics of this system, I just wanted to give a little bit more background about this. So this is actually data that from 2013. So at a place where I got a lot of my, did a bunch of studies this past year. And this is looking at the different genotypes and just the frequency of the different phenotypes that we observed in the field. And then I could do a quick little Hardy-Weinberg expectation from that. I hope you can see the distributions aren't too different. They're not significantly different. And so essentially these throat color phenotypes are potentially segregating in a way that we'd expect if it was a simple Mendelian locus. In addition, Barry has done field pedigree studies and breeding experiments to show that these alleles are highly heritable. So I'll touch more on the genetics later on. I'm trying to figure out actually what this OBY gene is, and I'll describe that at the end of the talk. But just to get back into the speciation aspect of things, this is a scenario I want you to envision in terms of, okay, we have these different lizard morphs. Maybe they have some sort of adaptive landscape where they differ in body size and territorial behavior. This is sort of fictitious data, but they're on different adaptive peaks. What would happen if we take one of the players out? I would argue that the adaptive landscape has to change because many of their traits are due to competition among each other. You can no longer play a rock, paper, scissors game, and therefore you must play some sort of other game. And so, in this sense, we might see changes in traits resulting from this. So, just to jump back into the hypotheses, I promised I would cover them. So this first one is just saying, okay, if you see monomorphic lineage or lineages that are fixed on one particular type, these should resemble the morphs that are found in these orange, blue, and yellow populations. And it doesn't have to happen this way. So derived lineages could have no throat color, just a white throat, or they could have some sort of novel coloration. And this tied into one of the big aspects of my project that I really enjoyed. I got to study the geographic variation of the mating strategies, which really hadn't been looked at much at all before I came into Barry's lab. He had been studying one population for maybe like 20 years, and so I was interested in what was going on in the rest of the range. And so I sampled 41 populations for these, for their phenotypes and scored what we presume are OBY alleles. And the sampling was a really big issue because it's fairly easy to detect if you see all the colors, but it's pretty difficult to say whether something is not there. And so I had to think about sampling. And so what I aimed for was a sample size of 30 lizards or greater, and I attained that for the majority of my populations, and this would give me a 95% probability of detecting a allele if it was as low as 5% frequency in the population. Another thing was just in terms of the vast majority of my populations had a sample size of 20 or greater, which would give me a 99% probability of detecting alleles at 10% frequency. So I tried to pay attention to the fact that, there's a sampling problem here. In addition, I resampled 25 populations to verify presence or absence, especially like if I was saying a population was missing a morph, I tried to go back there to make sure about that. Because these things are cycling over time, there's seasonal differences in how the colors are expressed and things like that. And in total, my data set was 1,694 lizards, including that nice orange one over there. All right, so here's the big picture of what I found. Just to orient you a bit, this is the range of the side blotched lizards. So they're found over most of the western United States and much of Mexico. These different colors on the map are the ranges of putative subspecies. And then this gray area was denoted a contact zone. And I'm going to be showing data on the different populations along the side of the map. So populations in this green area are shown on this green area on the side. And then the frequencies of the different alleles that I observed in males and females. In this case, this is Barry's study population in Los Baños, California, are shown here. And females also have the same color polymorphic alleles. That's a whole separate story as well. So what did I find? First of all, I found that the polymorphism is actually really widespread. It's not restricted to just a small part of California. So most of the populations in this green area were polymorphic for all three types, all the ones I sampled here in purple, and some of the populations in the southern part of this gray area all had all three types. And so it's actually a pretty common phenomenon for the side blotched lizards. However, you'll notice that the map is not completely filled in. There was some variation. And so there was populations where I was only able to detect blue and orange types. And so one is Anacapa Island in the Channel Islands. One is the Pisgah lava flow in Southern California. And then there's two islands off of Baja California that only have orange and blue phenotypes. And then there's some populations that are all orange. So the populations here in light blue adopt all orange phenotypes, and sometimes the phenotype is quite pronounced. This is an individual, I think, from up here in Washington. And then some populations in northern Utah are also all orange. So you can fix on this phenotype. And this was actually a surprise, which was there's some populations with a phenotype of blue throats with orange. And every individual has that. So all the populations here in red have that phenotype, as well as some populations in Big Sur have that phenotype. And this is the phenotype which I might describe earlier as what we would call an orange-blue heterozygote. But obviously something has changed in these populations. They are not segregating alleles anymore. And that's as much as I can say about what the genetics are going on there right now. But what I can say, sort of in summary, is all these populations do resemble the phenotypes that we observe in polymorphic populations, which is roughly consistent with this first hypothesis. All right. So moving on to the second hypothesis, if polymorphism actually acts to promote diversification, then polymorphic lineages should be ancestral. These monomorphic lineages should be derived. It doesn't have to be that way. Alternatively, these polymorphic mating strategies could be relatively new, and then the monomorphic lineages could say something about how mating strategies have built up over time. This is a phylogenetic question, and so to answer it, I sequenced two mitochondrial genes, one about 1,000 base pairs and one around 600 base pairs. And then I use maximum likelihood and Bayesian methods to reconstruct a tree. I'll show you the maximum likelihood one. And then I use the program Mesquite to reconstruct the ancestral states of the color polymorphism. So this brings me to, I think, my other big result to come out of my thesis, which was the evolutionary history of rock, paper, scissors mating strategies. And what I've plotted here are, these are the frequencies of the different throat colors at the tips. These are the geographic regions. These are bootstrap values. And then the branches correspond to different reconstructions of the polymorphism. And what you can see easily is that the ancestral branches are black, which corresponds to being trimorphic. And then towards the tips of the trees, you get these other populations. So populations that are fixed for orange are in orange, populations fixed for the phenotype of blue throats surrounded by orange are in gray, and then dimorphic populations are in light blue. And so pretty robustly, I think we can say that this rock, paper, scissors polymorphism has been around for a long time. And as potential further support of this, one of the closely related outgroup species are the tree lizards. They also have orange, blue, and yellow throat colors in some populations. And if this is a common ancestry, this would suggest that this polymorphism has been around for an exceptionally long period of time. And just to point out one other pattern here is that there's 8 independent losses across the tree. And in all cases, the yellow allele is lost. And so this is sort of an interesting example of convergent evolution here. This sneaker male strategy doesn't seem to do that well in terms of stability. And this is where I got my title in terms of rock and scissors beating paper. So in terms of thinking about why the yellow allele might be lost, and a natural first thought about why this could be is that the yellow allele could be rare in a population. However, it doesn't seem to be particularly rare. So looking at the allele frequencies at Barry's study population over a long period of time, the average frequency of the yellow allele was 0.4, and we can calculate a p-value about just by random draws about whether it's likely that you'd have 8 independent chances and have it all be yellow. So it's pretty unlikely. If I look across all my trimorphic populations, yellow is still about even with the other types. And so it's also relatively unlikely that this could all be due to chance. And so this is one possible thing that could suggest that selection could be involved, especially in some of these. I'm not going to rule out drift entirely, but selection seems like a reasonable thing to infer. But just to sum up, all of the reduced morph lineages of dimorphic or monomorphic were derived from a polymorphic ancestor, and there's been repeated loss of the sneaker morph. And what I'm going to ask next are what are the consequences of this morph loss, and this gets back to this hypothetical adaptive landscape. If you have rock, paper, scissors strategies, you take one out, what happens? So this was actually partly motivated by natural history observations in the field. At Barry's study population, you actually have pronounced male bias sexual dimorphism. So this is a male, this is a female. And for small lizards, this is a big difference. And actually, all trimorphic populations have male bias sexual dimorphism. But when I went to some of these populations where they've fixed on morphs, things are different. So there's some populations in this red area that males and females have equal body sizes. And there's even a population here in northern Utah where the females are larger than the males on average. So this is interesting variation within a single species in terms of sexual dimorphism. And how I addressed the evolution of this trait was I used a program called Brownie to compare 2 models for the evolution of male size, female size, and sexual size dimorphism. One was a single rate of phenotypic evolution across the phylogenetic tree that I showed you earlier. And I compared that to a model which had 1 rate for the trimorphic populations and another rate of evolution for the monomorphic and dimorphic populations. And just to summarize that result, the 2-rate model was conclusively supported for all 3 body size metrics. And I have a graph that shows these results in hopefully an intuitive manner. So this is looking at sexual size dimorphism, male size and female size, and then the relative rate of evolution that Brownie reconstructed. And this relative rate is the rate of evolution in the monomorphic and dimorphic populations divided by the rate in trimorphic populations. And so all these are positive, suggesting there's faster evolution in monomorphic and dimorphic populations for these traits than in trimorphic populations. So for sexual size dimorphism, the rate of evolution is 10 times as high in monomorphic and dimorphic populations, and even higher for male and female size. And so this, to my knowledge, in terms of West Eberhardt's theory, hadn't really been tested before. And so finding evidence of rapid evolution when morphs are lost was actually a cool new phylogenetic way of addressing this hypothesis. So lastly, could these changes that I've observed among populations actually promote species formation? So this brings me to the idea that potentially these populations could develop reproductive isolation. And I was trying to think about some ways, well, why would you get reproductive isolation from changing the number of morphs? One is that if you get rapid evolution among populations, this could potentially speed up the formation of incompatibilities among populations. Traits that are competitive in one social environment might have low fitness in another environment. And then you might have some sort of female preference or mate choice among populations for phenotypes that are particularly good in a social environment. So to answer this question, I went to a set of populations in Southern California, and one of them is dimorphic and one of them is trimorphic. This is the Pisgah lava flow, and on the lava flow you have lizards that can get quite dark, and right off the lava flow the lizards are much lighter. And much of this darkness is actually due to plasticity, though there's some, the ones that live on the lava remain much darker even when you have them off the lava for a while. So on the lava there's 2 morphs, off the lava 3 morphs. The lava flow is only about 22,000 years old, so it's formed rather recently. There don't seem to be any physical barriers to gene flow among populations. And so many models of speciation would predict no reproductive isolation in this case. There's been little time or isolation among these populations. This idea about polymorphism leading to new speciation, you might have noticed that I sometimes have it listed as morphic speciation just as a shortcut. This predicts that potentially we could see reproductive isolation at this area. And so I set up some genetic crosses among lizards, and so I took males and females from both of the populations and set up a breeding experiment so that a lava male was mated to a lava female, as well as an off-lava female, and then an off-lava male was mated to an off-lava female, as well as a lava female. And that way I could compare within versus between population fertility. And just to give you an idea about what a lizard breeding experiment looks like, these are breeding chambers that we constructed, and each one of these has 3 lizards, and then they have these central lighting sources. And my collaborator on this project, Leslie Lancaster, is checking one of the lizards to see if it was gravid with eggs. And in total, I had a sample size of 6 orange males, 14 not orange males, 17 lava females, and 19 off lava females in the breeding experiment. And as a result of that, I collected a lot of reproductive data to try and figure out what was going on. Just in the interest of time, I'm going to concentrate on the number of fertilized eggs, which showed the most pronounced effects. But I'd be happy to talk about the other measures if you're interested. And so here's the gist about what I found. So there was significant effects in a model for in terms of number of fertilized eggs of the population of the mother, as well as the morph of the father. And so in terms of looking at the population effects, here we have lava females and off-lava females, and then we're looking at number of fertilized eggs on the y-axis. The off-lava females, whether mated inside or outside their population, didn't differ in fertility, whereas the lava females, when they're mated outside their population, they have reduced fertility compared to when they're mated within their own population. And this is actually pretty substantial. They go from laying about 4 eggs to laying about 2 eggs on average. In terms of the effect of sire morph, we have not orange and orange males. The not orange males, whether mated inside versus outside their population, didn't show strong well, it didn't show significant differences, whereas the orange males, when mated outside their population, tended to have reduced fertility. So just to summarize those results, one of the things I think of in terms of this reproductive isolation, it's not complete reproductive isolation, it's just reduced fertility, but it's fairly substantial. And the fact that it's built up so rapidly in terms of 22,000 years, I think suggests prezygotic isolation. And this is further supported by looking at some of the other metrics I mentioned. When mated outside their population, the females actually produce more unfertilized eggs. And this suggests that there's something to do with either cryptic female choice or potentially like sperm egging compatibilities or things like that that's going on here. This really deserves more work to figure out what the mechanism is. The reduced fertility of these the outcross Pisgah lava flow females might suggest some sort of selection for assortative mating due to local adaptation. The Pisgah lava flow is a small isolated population that's surrounded by other populations on different habitats, and so gene flow, if there are adaptations for the lava flow, such as darkened color or a different mating system or something like that, they may want to maintain whatever those adaptations are from gene flow from outside the lava flow. And lastly, this observation of reproductive isolation is consistent with the predictions of polymorphism potentially leading to speciation, but this isn't to say that other factors such as ecological selection aren't likely to play a role as well. To do that, we'd need lots more breeding experiments and things like that tease those things apart. But this is an interesting case where, at least my initial reason for being interested in this population was the difference in morphs, and indeed they showed some reproductive isolation. But it would be great to go back here and do some more work. So just to wrap up this first part of the talk and think about the broad significance of this idea of morphic speciation and getting back to all these color types in other species, why is this interesting? Is that what I think is an interesting change in perspective about speciation is that it suggests that phenotypic diversification that's relevant to speciation can actually occur before geographic isolation. This is not to say that allopatric speciation is not important, but it's to say that essentially that sympatric morphs could be the starting material for new allopatric species. And so a lot of the focus on polymorphism in the past has been, okay, these things build up in sympatry, can they be torn apart by sympatric speciation? That can be fairly difficult, but perhaps an easier model to happen is that in sympatry you can get strong competition driving the evolution of morphs, and then across populations, these can evolve in new ways and give rise to species actually relatively rapidly. Another thing that I think it can help explain is sort of a larger phylogenetic pattern about why you see monomorphic lineages that resemble specific polymorphic types popping up in a phylogeny when they're closely related to a polymorphic species. This might help explain that polymorphism can sort of give rise to these monomorphic lineages. And lastly, I think some of these ideas offer new ways to predict the occurrence of both rapid phenotypic evolution and the formation of reproductive isolation that haven't really been considered before. So that's sort of the nutshell of my thesis. Hopefully all that sounded interesting because it's led to where I am now in terms of delving back into the system. I thought my PhD project was great. I got to travel around a lot, got to look at lots of lizards. So I wanted to come back, and so it's great to have the opportunity to do so here. So what I'm specifically working on now is trying to locate the OBY locus that gives rise to the morphs that are found inside large lizard. And the significance, I think, of this is that this is a gene with major effects on development, on behavior, on physiology. These morphs are really distinct in coloration and testosterone levels in a whole suite of traits. And so this could tell us something about the genetic underpinnings of those phenotypes. I think its discovery would also allow a really detailed look at how selection in the wild then affects genetic variation. So Barry's been studying this population for years and years and years and measuring selection. And an easy prediction from that work in terms of they're playing a rock, paper, scissors game that maintains variation over time, is that you should expect to see a very strong signature of balancing selection on whatever gene this is. And then another thing is that there's fairly little that's known about the genetic basis of sexually selected phenotypes. There's a number of researchers that have made a great deal of progress on looking at naturally selected phenotypes, and this has been a great boon to evolutionary biology, but I think that sexual selection deserves its due as well. So the strategy for trying to find this gene is based on techniques that are being developed here in the museum, and specifically on sequence capture techniques. And so to start off, I've sequenced RNA to gather data on the coding sequences for the expressed genes in the side-blotched lizard. And so far I have transcriptome data for 11 individual lizards, which includes representatives of all the homozygous types. And then there's a lot of bioinformatics of assembling the sequences and annotating them. This is something that I just completed, and part of the reason it took a long time was I did multiple rounds of quality control because, in essence, there may be just one gene or two tightly linked genes, and so I need to make sure that I get whatever gene that is in my data set for future steps. And in total, I now have 16,345 contigs and 28.7 megabases of sequence data, which may not make that much sense to you. But just to put it in perspective, the one lizard genome that's been sequenced is the anole, and when they reported in their paper, they reported a little over 17,000 genes. And I'm not arguing that all my 16,000 contigs are separate genes. They might be separate parts of the same gene, but for the most part, I think they're unique genes. And so I actually have a fairly big representation of the coding sequence for side-blotched lizards, I believe. The next step of this is to use sequence capture methods to collect genetic data for a large number of individuals and scan for genetic variation. And I plan to perform this in December. I have to wait to get the sequence capture kit. And then there's going to be, I have to assemble the sequences and identify genetic polymorphism. And finally, I'll be able to test for associations between these color morph phenotypes and the genotypes in the genome, and hopefully find some sort of correlation between these two. And just to think about what that would look like, should I be successful, I think it would be interesting to know what this gene is for many reasons, but I have big plans. So one of them is that I could potentially look at sequence variation in this OBY gene and look across the populations that I showed you earlier that differ in number of morphs. So I could go to an all-orange population, and what I would predict is that whatever the orange allele is should be fixed in those populations. And then I could also test if this gene is variable in other species that show color polymorphism. For instance, the tree lizards that also show similarities and throw color to the side-blotched lizards, to try and see if the same genetic basis gives rise to polymorphism across species. We really don't have much of an idea at all about the genetics of these distinct polymorphic types, and so being able to get into that would be pretty interesting. However, there's no guarantees that I'll find this gene. At the very least, I hope to localize the region through linkage, but there's lots of other things that I can do with this genetic data. And so, for instance, I could test whether morphs within each sex show pronounced differential gene expression. This is one thing that was done in dung beetle morphs, and they actually showed that some of the male types show more pronounced differences in gene expression than that found between males and females. So this is one interesting thing. Another thing would be to try and identify the downstream genes that give rise to the different color pigments. So this OBY gene, I think, is something upstream that triggers a whole series of differences in these things, including behavior and coloration. But we know fairly little about what gives rise to little lizard coloration, except for things like MC1R. And so one of the things that I did in my data was I sampled pigment on the same lizard, or skin on the same lizard of a pigmented area and a non-pigmented area, and so I can compare the expression between those two areas. And then, given I have all these genetic markers, I think I could do a lot of interesting population genetics. So for instance, I could go back to the Pisgah lava flow and do some sort of hybrid zone study and look to see if any of those 16,000 genes shows differentiation or reduced gene flow. And lastly, I could look for, say, signs of population bottlenecks in populations that have lost morphs to try and get at what's going on. Why do populations lose morphs over time? And I'd also be happy to talk with anyone else about ideas about what I could do. I think that there's a lot of possibilities here, and so I'd love to talk with people further about that. So I'm going to end there, and except for acknowledging all the help that I've gotten with this project. I had a huge amount of help during my PhD on this project. Lots of field help, lots of lab help. But since people here are in the audience, I want to make sure to thank Barry Cinervo, my advisor, Rasmus Nielsen, and KV, who have been great helps with this project. And with that, I'd like to take any questions. Thank you.